Hi, good morning, everybody. Thank you for amazing talks and discussions today. So I will be talking about my own thesis project, and basically, which is on the resting state functional connectivity and white matter integrity as biomarkers to track brain changes in former athletes with multiple concussions. So first, I will be talking about the background of the study. Then I will move on to the hypothesis, methods, results, limitations. I will touch upon future directions of the study, and the questions will be taken up at the end. So the study background, just very briefly, I wanted to mention that there's approximately 10 million of cases per year of traumatic brain injuries, and the vast majority of these are mild traumatic brain injuries. Even though most people recover from these, certain portion of people do go on to develop chronic traumatic encephalopathy, which is believed to be the progressive neurodegenerative disease associated with repeated concussions. So there's currently no antemortem biomarkers for CTE, and the diagnosis can currently only be done postmortem on the postmortem brain tissue. And I know we've already touched upon the pathology of CTE before, but just to briefly mention, it's characterized by abnormal perivascular accumulation of tau in neurons, astrocytes, and cell processes in an irregular pattern at the depth of the cortical sulci. And then looking at the retrospective cohort analyses for the CTE diagnosed participants, we can note that certain symptomology comes up more frequently, which is the cognitive issues such as memory, executive function, and attention deficits, as well as the behavioral and mood changes such as impulsivity, aggression, and depression. So the CTE incidence among the literature is currently undefined, and it varies quite a lot actually between the studies. So going from 87% within the players of the American football to approximately 33%. And we've mentioned today the study done actually here at the University Health Network currently has the CTE incidence in 22% among their cohorts. So it's quite a lot of variability within. So just moving on into the modalities implemented as a part of this study, we're using the resting state functional MRI, which is basically in a direct way to measure functional connectivity between the different areas in the brain. And what it does, it measures the blood oxygen level dependent signal synchrony among different brain areas. So basically, if there's an increase in blood oxygen level dependent signal in multiple areas at the same time, it's believed that they're functionally connected and basically working at the same time. And the resting state, meaning the subject is at rest and not actively participating in any cognitive tasks. So this slide just to signify that different symptomology associated with CTE can be associated with different functional connectivity networks within the brain. But specifically in concussion, what comes up a lot is the default mode network, and actually the saliency network, which has been found to be altered in the traumatic brain injury participants, which is what we'll be talking about right now. So saliency network, if you're thinking about in your own environment, every time there's a lot of different stimuli which are competing for our attention. So what saliency network specifically allows us to do is to pay attention to the important stimuli to guide our behavior and for more in-depth cognitive processes. And actually, saliency network has been found to be altered in the depression, Alzheimer's disease, and post-traumatic brain injuries, which is why this is one of the networks that we are paying closer attention to. Now, we've been talking a lot about DTI, so the diffuse tensor imaging today. Just to kind of give you a quick run-through, what I will be talking about as a part of this presentation is the measure of functional anisotropy. So if you think of white matter tracts as a highway, basically what DTI does, it tracks the water molecule diffusivity. So if you think of white matter tract as a highway, if it's an intact white matter integrity, that means the water molecules will diffuse all in the same direction, as you see here on the left-hand side. And then the values closer to 1 on the functional anisotropy scale would mean intact white matter integrity. However, if white matter integrity within the tract is compromised, that means the diffusivity of water molecules can be all over the place, right? So then the lower values on the functional anisotropy scale would mean compromised white matter integrity. So in terms of DTI, once again, just going back to the symptomology from the retrospective CTE studies, there's a lot of evidence from other cohorts that certain tracts are responsible for certain symptomology. However, looking specifically in concussion, corpus callosum has been the one mostly studied, and it has been shown actually to have decreased white matter integrity comparing to other populations, and it has been associated with worse cognitive symptomology and the higher number of concussions. So the aim of this particular study is to assess the use of resting state functional connectivity and white matter integrity as biomarkers of longitudinal changes in a cohort of former contact sport athletes. And then just going quickly onto the methodology of the study. So the recruitment is still ongoing. The inclusion criteria are participants under 85 years old who are fluent in English and are former professional athletes in a contact sport. And very important to mention, the participants with dementia and any ongoing neurological and psychiatric issues have been excluded from the study. So the biomarker collection includes the baseline assessments and the 2-year follow-up, and includes the magnetic resonance imaging for the anatomical structural scan, DTI, as well as the resting state functional MRI. Full neuropsychological, neurological, and neuropsychiatric assessments were completed, and cerebrospinal fluid and blood collection was done. And cerebrospinal fluid collection was done to assess for the presence of Alzheimer's disease pathology in this cohort. So just to quickly mention, these are some of the cognitive and memory and behavioral domains that we were assessing, and just a brief overview of some of the tests we have implemented. Just for the purpose of this presentation, I will be talking about the trail making test part B, which is a measure of the executive function, as well as the personality assessment inventory, which was a measure of depression as a part of this particular presentation. So these are the white matter tracts that were segmented. The one that we will be paying a closer attention to is once again corpus callosum, and it is responsible for the interhemispheric communication in the brain. So the data analysis was done using the FSL and CON toolbox, and just going into our results. So we currently have 25 former contact sport athletes with the demographics as you see on the screen. One of the things that is important to mention, as you can probably tell, the concussion number, the minimum concussion number was 0. That's because one of the most prominent symptoms of a concussion would be memory loss. And because these are all high performance contact sport athletes, we do believe that even though these are self-reported, that they do definitely have a certain number of subconcussive or still concussive blows that they might not necessarily recall during our self-report assessment. So just going through the results, what we found actually is the decrease in the functional connectivity within the salience network for the 2-year span in this cohort, and it has been seen over a variety of different nodes within the saliency network. And actually, the connectivity, the functional connectivity within the salience network was actually correlating with the worsening depression in this cohort over time. And then looking into some of the other tracks, so right executive network was also associated with worse scores on the executive functioning tests. And then going into white matter integrity, as I already mentioned, so the corpus callosum was found to be decreased in white matter integrity values in the concussed population compared to healthy controls. And what we see in our study is actually there's a significant decrease in white matter integrity in corpus callosum, both on the functional anisotropy and medial diffusivity values over the 2-year span. And the worse integrity at the baseline actually predicts worse executive change over the 2-year period. So then the limitations of the current study is because we are implementing resting state fMRI, and the brain default energy states is related to the mental state. So we unfortunately cannot control for the mental state of our participants. So, and it could actually change depending whether the participant is tired or excited, as well as we do have a highly varied study cohort. So as you mentioned, the number of concussions varies quite a bit between the participants. And going into the future directions, I'm planning to do a direct comparison between different neuroimaging modalities, as well as the biofluid analysis, such as blood and cerebrospinal fluid, and then compare it to the pathology that we can find upon the autopsy. And another thing that we do have on these participants is the PET-tau imaging. And just to briefly mention that, by looking at the longitudinal differences in the PET-tau imaging, there are actually people who are increasing in terms of this indirect measure of tau pathology in the brain, and the ones who stay approximately the same. However, we're still recruiting more participants for that, which is why those results were not included as a part of this presentation. However, one of the future directions would be to see whether, what other differences, underlying differences that we can see in other neuroimaging modalities between the people who are accumulating this underlying pathology and the ones who are staying the same on the measure of the tau. So I would like to thank study participants for volunteering their time for this project, as well as funding, and our collaborators, and as well as my amazing lab members. Thank you very much, and if you have any questions.